So I think that means we're good to go, so we can get started. Welcome everyone who's joining us today. My name is Morgan Einesen. I am the Manager of Education at Fighting Blindness Canada, and we're really excited to have you here for our Facebook Live today, all about our new Transformative Research Awards. For nearly 50 years, next year is our big anniversary. Fighting Blindness Canada has helped move the needle forward on vision research, underpinning our understanding of the genetic causes of eye disease and contributing to the development of potential sight-saving treatments. However, we know that there is a lot more work still to be done and more to discover. And that's why we're thrilled just last week in our e-news to announce that we are providing $1.67 million in funding to 2 Transformative Research Awards, which will further push the boundaries of vision science. So today I am joined by Fighting Blindness Canada's Director of Research and Mission Programs, Dr. Larissa Moniz, and we're going to just talk a little bit more about these exciting projects. And if you have comments or questions, please feel free to add them in. I will be monitoring them throughout the session. So Larissa, thanks so much for joining me on this very rainy Monday. Could you start off and tell us about why Fighting Blindness Canada decided to offer these new awards?
Absolutely. I'm so excited to be able to talk about this. So this has been, as you'll hear, it's been quite a long process of putting together the awards, going through the review process. And so it's really, really exciting to be able to talk about them because they're both really wonderful and exciting projects. So what is different about these awards than the other awards we give out? So in the past, the primary type of award that Fighting Blindness Canada has given out has been what we call our research or operating grants. So this is approximately $100,000 for a year for like 2 to 3 years. And these awards are been really, really important for the research community. They help provide almost seed funding. So researchers can start to develop some early research ideas, test out hypotheses, and then hopefully use the data that they've collected to then apply for larger awards. So some of these multi-year awards from organizations like Canadian Institute of Health Research, which is one of the largest funders in Canada. And so the research grant awards, they have been and will continue to play a really important role in our funding structure because it really helps us allow researchers to test out these new ideas and potentially high-risk ideas. However, there is a limitation to the funding because it is sort of limited in a little bit both in terms of money and time. When you think about how much time and money it takes for a research project to develop, this really only can fund a little bit of that. And so we were inspired by the success of another recent award program that we ran. It was called Restore Vision 2020. We invested about $2.5 million for projects to help develop therapeutic avenues for advanced retinal degeneration, including retinitis pigmentosa. And what we found was a couple of these projects, with funding, including funding from Fighting Blindness Canada, it really helped them move through the preclinical phase. And one of them is currently in clinical trials with a product very similar to what was funded by Fighting Blindness Canada. And another one, which is about stem cell therapy, is in very late stage preclinical development, hoping to move to clinical trials. So we really wanted to try to recreate a research program like this that is offering larger scale, longer term funding for projects that already have some of this preliminary data. So they sort of know that the root idea is good, that it is strong, and this funding will hopefully allow them to sort of ask the bigger questions, accelerate the pace of their research.
So taking these ideas and being able to move them forward, which is so critical. Mm-hmm. So can you take me through the process of how this all worked? How were these 2 awardees chosen?
Yeah, so it was sort of both similar to some of our general, what we consider a really robust review process, but with a few extra steps built in because these were very large awards for us. So for this first round of the Transformative Research Awards, and we are hoping this is just the first round and that in future years we'll run this competition again and award even more really innovative, exciting awards. But for this one, we invited researchers to submit proposals for research that was ambitious, that was innovative, and that would specifically advance the development of new therapies for retinal degenerative diseases. So that could be age-related macular degeneration, inherited retinal diseases, or glaucoma. And we opened up the competition. So it was for discovery or basic research, translational research, or even clinical research if that came in. And we also know that research is very much a global endeavor. And so we also opened up the competition beyond Canada. So it was open to researchers from around the world. And we put out the call and we received 21 submissions from world-class research groups in Canada, in the US, in Europe, in Australia. And then we basically created a short list of the top proposals and invited them to submit a full application. And then this was reviewed by a really rigorous review panel, which had experts from around the world, and each application was discussed. And based on the panel, the scientists' recommendations, their discussion, we were able to, at the end of the process, fund the top 2 applications. And the 2 applications that we are funding is Dr. Marius Weffing from the University of Tübingen in Germany. He will be developing a neuroprotective drug therapy to help reduce photoreceptor cell death and slow vision loss in inherited retinal diseases. And we also are funding a grant from Dr. Luis Alarcon Martinez, and he's working out of the Center for Eye Research in Australia. And he's using some really cutting edge imaging technology to understand how, when blood supply is disrupted in glaucoma, this may cause optic nerve damage.
I just popped up a slide here that has photos of both of these researchers as well as their institutes. So I'd love to dig in a little bit. Oh, did that go away? I think it went away already.
That's okay.
We'll get that back up there. There we go. So I'd love to dig in a little bit more to each of these projects. So in the project that's being spearheaded by Dr. Weffing, they're investigating a new therapy that is gene agnostic or gene independent. So could you tell us a little bit about what that means?
Mm-hmm. So their therapy is focused as a gene agnostic approach for treating inherited retinal diseases. And inherited retinal diseases, they are this group of eye diseases that includes diseases such as retinitis pigmentosa, Usher syndrome, Leber congenital amaurosis, as well as many more. But all of these diseases, they cause progressive vision loss that ultimately leads to blindness. And in all cases, the death of photoreceptor cells, which are the light sensing cells in the eye. This is the key event that leads to vision loss. And as the name suggests, inherited retinal diseases, these diseases are inherited and they are caused by gene mutations. And there are over 300 different genes that can be mutated to cause an inherited retinal disease. So a lot of the current therapies that are being sort of tested in clinical trials, as well as the first approved treatment for an inherited retinal disease, which is called Luxturna, these are gene specific. So what this means is they are either providing a functional copy of the gene back into the cells to replace the mutated gene, or even potentially try to fix that mutation. So these are really, really promising, but there are both technical challenges as well as the cost and time involved if you're trying to make a gene-specific therapy for every single type of gene mutation. So for example, there are about 22,000 Canadians who have an inherited retinal disease in Canada, but only 50 Canadians are eligible to receive Luxturna. So if you can imagine that if we had to make a special gene therapy for the other 19,950, it just can be very expensive, take a long time. So in addition to some of this gene-specific work, there's also a lot of researchers who are looking at more gene agnostic or independent approaches, and this means it'll be a treatment that could be potentially a treatment for people regardless of what mutation they have. And that's sort of what Dr. Weffing is trying to do. He's trying to solve a problem that occurs in some inherited retinal diseases that's about a process that's called protein folding.
It's so interesting to me, these gene agnostic approaches. And if you're more interested in learning about these approaches, we had a viewpoint last year that was all about gene agnostic therapies, which I definitely recommend. Can we talk a little bit more about this protein folding process? So how does this play a role in the cell, and what is Dr. Weffing using this award to do?
So in the cell, there are thousands and thousands of proteins. And proteins are essentially, I consider them sort of like the little worker bees in the cell. So each protein has a very specific function. So in a muscle cell, there will be special proteins that help the muscle cell contract. In a nerve cell, there will be different molecules that help the nerve cell sense things. And in a photoreceptor cell, there are special proteins that help the photoreceptor cells sense light. So each protein is folded in a really, really specific way, and that specific structure is really important so that it can do its job properly. The problem is that sometimes when you have a gene mutation, it causes the proteins to not fold properly. And you can sort of imagine a folded protein. If you imagine it like a ball, like a tightly wound ball of yarn, I feel like this will be an analogy close to Morgan's heart, who's an avid knitter. So it's nicely compact. You can knit very easily, but if you imagine that you had this ball of yarn just like opened up and then it was in a big bag with lots of other balls of yarn that were also not rolled up, you can imagine that they would just clump together, get tangled up, becomes a little bit of a mess there. So when the cell senses this sort of clumping of the proteins, it basically sets up this an internal alarm system. And the cell then starts to use a lot of energy to try to fix this problem. So in an inherited retinal disease, this might happen. And then so the cell's already struggling a bit. It's got the gene mutation. It's maybe struggling to do its normal functions. Maybe it's a bit damaged. Then you have this protein folding problem. And the cell's like, oh God, all of a sudden the second alarm system is like, oh my God, we got to fix this problem. And it starts to use a lot of energy to try to fix this problem. So it diverts energy and it can potentially just run out of energy, reach a crisis point and die. So to me, I sort of imagine it like if you have a fire alarm that's a little bit too sensitive, we all know it's really important to have a smoke alarm in the house. But if it's going off every time you're creating toast, it's just like, a bit of a pain. And that's sort of what's happening in an inherited retinal disease cell. It's sort of focusing too much on a problem when it has bigger problems to worry about. So in this case, Dr. Weffing is trying to develop a new neuroprotective drug that's going to sort of put a brake on this energy-intensive alarm system, sort of telling the cell, you can slow down, you don't have to worry about this protein folding problem, you have bigger problems to worry about. Helps the cell preserve some energy. And there's been some preliminary work that has shown that this drug can actually reduce photoreceptor cell death in some animal models of retinitis pigmentosa.
Well, I always love a yarn analogy, and I will tell you there's a technical term for that, and it's called yarn barf. if anyone ever wanted to know, that's what it's when it's all just like everywhere. So it sounds like a really fascinating project. So what are the potential impacts of the work that could come out of this?
So there's a few different things. So with the funding from this Fighting Blindness Canada Transformative Research Award, Dr. Weffing is going to be working with a whole team of international collaborators. And until now, they have been working with sort of an early version of the drug. We can call it a beta version or a version 1.0. And what they're trying to do is identify an optimized version of the drug. So sort of a version 2.0. This version will hopefully be more effective, more suitable for clinical use in humans. And the second thing they're trying to do is they're going to be testing out if the drug is effective at preventing photoreceptor cell death in many different types of inherited retinal diseases. So they've tested it out in a few sort of mutation models. So they want to test it out in many, many more, like 12, 15 different types to sort of see, is this a treatment that could be used by many, many different people? And so we're really hoping that this will contribute to some very important preclinical work that might help move this potential therapy a bit closer to clinical trials if it is successful.
That's great. So really testing to see if it is gene agnostic, it could apply to multiple different conditions and mutations.
Absolutely.
Before we move on to the second project, we do have a question from Ken. And Ken was wondering if the competition included prosthetic or other supplemental devices.
So it was open to research about prostheses, optogenetic, supplemental devices, because all of these could be considered as therapies or treatments for retinal degenerative diseases. In the end, the two sort of top ranked ones weren't about prostheses, but it was definitely open to this area of research.
For sure. So the second project, as you mentioned earlier, is about glaucoma. And you mentioned that Dr. Alarcon Martinez will be trying to understand how disrupted blood supply can cause optic nerve damage in glaucoma. So can you talk a little bit about the relationship between glaucoma and the blood supply in the eye?
Yeah, absolutely. So for anybody who maybe doesn't know, glaucoma, it's the leading cause of irreversible blindness worldwide. So it has a really, really large impact on people around the world. And in glaucoma, vision loss occurs because of damage to the optic nerve. And the optic nerve is the part of the eye or the cells in the eye that send visual information from the eye to the brain where images are made. And so traditionally, we think about optic nerve damage as being caused by high eye pressure in the eye. And so you often, if you have glaucoma, it will often first be caught because you have high eye pressure. And currently all the treatments around are to lower pressure. So there's drops, there's surgery, and that's the only treatment strategy we have. However, this probably isn't the full story because even with these treatments, they aren't always successful. And we know that optic nerve damage can occur even when pressure is under control. So there has been for many years this sort of discussion among scientists and clinicians about does blood supply play a role in this? And as you can imagine, blood supply, it's really important for all parts of our body. Blood helps transport oxygen, it helps transport nutrients around the body, and that's important for all of the cells, including the optic nerve cells in the eye. And clinicians have known for a long time that there is essentially a disrupted blood supply when patients have glaucoma. However, it really wasn't clear why this disruption was happening. And it also wasn't clear if it was a cause of the optic nerve damage, if it was causing the glaucoma, or if it was just a symptom. So maybe like it was there, but what was like wasn't important actually. So in a very recent breakthrough, Dr. Luis Alarcon Martinez, he was working with Dr. Adriana DePaulo at the University of Montreal. They discovered that there were these small tubes called nanotubes, which connect blood vessels that regulate the blood flow in the eye. And so they showed that these nanotubes break in some eye conditions, including glaucoma.
That's such a great Canadian connection there with Dr. DePolo at the U Montreal. So with the funding from this award, what is the research team going to be looking at?
Yeah, so this sort of study with Dr. Alarcon Martinez and Dr. DePolo, as well as other investigators, this was really the first time a potential link between blood supply dysfunction and glaucoma had been identified. So what the researchers want to do now is they want to sort of tackle what has been a very long unanswered question in the field. And so they're going to be using this really, really cutting edge imaging technology that helps them see these nanotubes that connect the blood vessels. And they really want to try to understand better about how is the blood supply disrupted, specifically in glaucoma. They want to identify some of those like molecular players or proteins that could be responsible for the nanotube breakage, for the blood supply being correct. And they're also going to ask an important question. So if in an animal model of glaucoma, if they can manage to preserve the nanotube, so make sure the nanotubes don't break, does this actually improve blood delivery? And most importantly, does this reduce optic nerve death and preserve vision in an animal model?
So fascinating. A really interesting project. I can see why these two came to the top. Really interesting. So what are the implications, the broader implications of this work around glaucoma?
So I think, so this work is what we would call either discovery or basic science. And I think what is really most exciting about this project is it actually has the potential to provide a completely new fundamental understanding of why optic nerve damage is occurring in glaucoma. Just it could really change how we think about the root causes of glaucoma. And that also means that once we understand that, it might help us identify new ways to treat glaucoma. It doesn't mean that we would stop doing pressure lowering drops or pressure lowering surgery, but this could be an alternative treatment that could maybe go hand in hand with some of the pressure lowering treatments that we know do have a lot of success. And I think what is sort of exciting even beyond the glaucoma field is these little nanotubes. It wasn't just the first time they'd been found in glaucoma, it was the first time they had been found sort of full stop with blood vessels. And so it really could increase our understanding of how we understand and treat other diseases where there is a blood supply dysfunction. And that includes eye diseases like age-related macular degeneration, diabetic retinopathy. It also includes diseases like stroke or Alzheimer's disease. So the impact could actually be really, really huge for diseases beyond glaucoma.
Wow, that's so exciting. Thank you for sharing about both of these projects. I'll turn it over to our audience to see if there's any other questions. I had another one, but I think you've kind of just answered it actually, and it was asking if there's a possibility that this research could help other eye diseases in the future, and it sounds like Absolutely. And there's so much
Yeah.
around like the optic nerve as well. Like, would this have implications for optic nerve conditions as well?
I think it could. So right now they're looking at glaucoma, but it is very possible that some other optic nerve or maybe even other eye conditions, as long as if there is an impact of blood supply, could also
Mhm.
be influenced by it.
Absolutely. I mean, yeah, the blood supply affects our whole body, so it's
Hmm.
very interesting to see what the broader
Yeah.
implications of this work
Hmm.
could definitely be. Let me just have one more look at the comments. Sorry, I can't see them on my screen, so I have to, here, you know what I'll do? I can take down these handsome faces now and see if I can get our comments up. All right, I don't think we have any other comments, so I think we can close off for today. Thank you so much for joining me, Larissa. It was really interesting to hear more about these projects. And if out there you're interested in learning more about these projects and see short videos from the researchers involved, you can visit us at fightingblindness.ca and sign up for our e-newsletter to keep up to date on the latest in vision research for these projects and our future research competitions. If you're interested in gene therapy, specifically, we have a wonderful Viewpoint webinar happening this week on Wednesday at 4 p.m. Eastern Time, where we're going to be talking about a lot of really cool new projects in gene therapy, gene editing for inherited retinal diseases and wet AMD. So I encourage you to take a look at our website and sign up for that if you're interested. And with that, we'll sign off for today, and hopefully we'll see you at one of our events very soon. Take care.
Thank you, Morgan.